A big part of Unit 1 is protein purification. And so it's true that before any proteins can be analyzed, we really need to purify it away from the other components of the cell. And so where do we get these proteins from? Well, it could be from a natural source, and that's, say, the protein's isolated from the tissue that it naturally occurs in at the natural levels that it occurs in those tissues. And so an example might be if we wanted to study the enzyme lactate dehydrogenase from cows, we could grind up their heart in a blender and extract the protein that way. We could also maybe grow up yeast or bacteria and go after the proteins that they're growing. A problem sometimes with natural sources, though, is the proteins that we might want to study could be in very, very low concentration and very difficult to purify to give us enough to do what we want to do. And that's where recombinant protein expression comes in. This process, we're going to add a gene that encodes a protein into some other organism so we can actually express that protein at very high levels. And so an example for this would be E. coli, where we can take the gene of interest. So maybe it's a bacterial gene we want to study, maybe it's a human gene or a mouse gene, and we want to study the protein product from that. We could add it to this small circle of DNA called a plasmid, and through a process of transformation, we can incorporate that plasmid into the E. coli. And what we will end up doing is exploiting the protein production machinery of that bacterium, of E. coli, so that we can ultimately produce the protein that we want. And so we can have the protein expressed by that cell. And this often can be accomplished where the protein is expressed at very, very high levels. So we will get a lot of protein out of this process. We're going to talk a lot more about this particular concept of recombinant protein expression, more so in Unit 2. But just know for now, what we're going to purify for Unit 1 is a protein that has been expressed through recombinant protein expression in E. coli. Now, in order to get to the protein, we do have to break open the cells, and that process is called lysis. And this can be accomplished a couple different ways. One is enzymatically, so we could use an enzyme like lysozyme to break open the cell walls and allow the cell to spill out its contents. And you probably talked about lysozyme in Chem 461. Could also accomplish this mechanically, so the example of cow hearts was an example of mechanical breaking. There are also other instruments that can be used to shear cells open mechanically. It's called a French press, but you don't want to be making coffee in it. There's also sonic ways to do this, where we can use sound waves to rupture the cells, and this is called a dismembrator. And we will utilize a sonicator for Unit 2 when we study those methods, but what we're going to use in Unit 1 is lysozyme to enzymatically break open our cells. In any case, what happens is we lyse those cells and we get what we would call a crude lysate. And this contains all the components that were in the cell, membranes, cell wall fragments, soluble proteins, insoluble proteins, DNA. All of that is in this thing we call the crude lysate. It's just cells that have been busted open. And that's kind of a big mess, largely because of the DNA. It creates a very viscous solution, and we need to shear up that DNA, and we'll talk about the way we do that later on. And what we need to do is centrifuge this so that we can get what we call the clarified lysate. The clarified lysate is the supernatant that we get after centrifugation. In our case, our protein will be somewhere in that supernatant, in that clarified lysate. Now, some folks might study membrane proteins or other things like that, and those would end up in the insoluble pellet down here. But for us, we're going to go after that clarified lysate, the supernatant. Once we have that supernatant, we can utilize selective precipitation to remove some proteins we don't want and concentrate our protein as well. So some proteins are more soluble at high salt concentrations, and we can use the concept of salting out or differential precipitation to perhaps salt out our desired protein or contaminants. And so the way this works is we use this salt called ammonium sulfate, and ammonium sulfate is very good at keeping proteins very stable. And if we have a mixed protein solution, and we add ammonium sulfate, we can selectively precipitate a protein that is perhaps a little less soluble than the others. And so we can centrifuge that and we can isolate that protein. If that's the protein that we want, then great. But maybe our protein is still up here in the supernatant. So we can then add additional ammonium sulfate, and maybe we want this newly precipitated protein, or maybe we don't. Maybe we want this other protein. So it kind of depends on the particular example. But these proteins can then be separated through this salting out technique. And one thing I want to point out is that this process can also concentrate the sample. So let's say we were after this protein right here. Well, now we can bring this up in a much smaller volume. The initial volume may have been very large, but we can bring up our precipitated protein in whatever volume we want. And indeed, we are going to utilize ammonium sulfate concentration or precipitation for both a purification technique as well as a concentration technique. I also want to point out that we're using ammonium sulfate for a very specific reason, because this salt stabilizes proteins. It reinforces the hydrophobic effect, and it keeps proteins folded up. So the proteins are going to precipitate, but they are not going to denature. We don't want to denature the proteins because then they wouldn't be active. So even though they form a milky precipitate that can be centrifuged and we get a pellet, those proteins are still in their native folded states. They're just in this form that is not soluble. That's in contrast to other salts. So if we wanted to denature proteins, we might use something like urea or guanidinium. Those agents cause proteins to denature, whereas ammonium sulfate causes proteins to stay folded up. Now, from there, we're going to utilize column chromatography. And chromatography in general, of course, is the separation of a mixture. Now, these techniques, these chromatographic techniques, can be carried out in a column with a bed of resin, and there are specific properties. And so this would be a column made of glass or plastic, and it has some kind of resin in here that has a particular property that could separate based on charge or size or something like that. We, of course, would have a buffer in here, and this is going to be very, very important as we're going along to ensure that we do not run our columns dry. We always have to have a buffer sample above what we'd call the column bed, above this line right there. So never let your columns run dry. Now, different components of the mixture, different proteins and small molecules will have different interactions through this if based on charge or size or whatever it happens to be. And those different components are going to migrate through this bed of resin at different rates. And so what we would do over the course of a purification is we would collect the protein that's coming out here in solution in different fractions. So we might collect every 1.5 mils or something, we would change a tube, and we would get a series of fractions, and we could generate what we call an elution profile. And so an elution profile is looking at the amount or concentration of a protein as a function of time or volume or fraction number, could be another option. In any case, we're looking at how the protein concentration changes what's coming out of this column as a function of time. And so in this example, protein A elutes from this column first with the least amount of time. Then protein B is in the middle, and protein C elutes from here at the longest time point, the highest fraction number. And that means that protein C was interacting more so with the column or was adhered to the column or something like that, whereas protein A flowed right on through, perhaps had minimal interactions with the column. So that's a general look at column chromatography, but of course there are specific examples, and one that we're going to use is ion exchange chromatography, and this separates proteins based on charge. If you're familiar with how your water softener works, this is sort of the same idea here, is that there are these resins and they contain inert beads that are made of something like cellulose or polystyrene or agarose or something that really is chemically inert, but they have attached to them a chemical group such as sulfonic acids or carboxylic acids. Those are going to have a negative charge to them. And so we call these cation exchangers, right? Cations are, of course, positively charged. And so our analytes, or the things we want to study, might have a positive charge. And so they would stick then to a cation exchange resin because the cation exchange resin has a negative charge. Anion exchange resins interact with anions because those resins are positively charged. Quaternary amines should make sense. That's going to always carry a positive charge. Diethyl aminoethyl will have a nitrogen with 3 groups, and at certain pHs, most pHs, it will carry a positive charge. So if we look at something like proparam, we can identify what the pI value of a protein is, and then we can select a buffer that will give, with a pH, that will ensure the protein has a particular charge. And then we can select an appropriate resin such that the protein will stick to it. Maybe we don't want our protein to stick with it. Maybe we do. It kind of depends how we set up the particular experiment. So in this example, let's again look at, we have a mixed solution of proteins here, and we're going to use an anion exchange column. Notice we have all these positive charged beads in there, and so anions will stick to it. And so if we load the column with our mixed protein solution, we see these things that go right on through the column. They don't interact with that at all because these are probably positively charged, or they could be neutral. They could have a charge of 0 and they're not really going to interact with that. So of course positive charges are going to fly on through because those are cations, and this is anion exchange. And so if we want these proteins, whatever these happen to be, these black ones, then we're good. We have purified that away. But maybe we don't want those. Maybe we actually want one of the proteins that's up here. And to get those off the column, to elute them, we could do a couple things. We could add very high salt. The high salt will interfere with the electrostatic interactions that are holding the proteins to the column. Or we could change the pH. If we change the pH to a value, in this example, below the isoelectric point, then we would cause those proteins to become positively charged, and then they would elute from the column. So there are various ways to elute proteins from an ion exchange column. In any case, we have definitely separated these two populations of these black ones and the remainder. And so ion exchange chromatography is a very powerful and useful technique for protein purification. And you will get to experience this in a few weeks. Gel filtration chromatography is also a very common technique, and this is often called size exclusion chromatography because it separates proteins based on size primarily, but also shape in some ways. And the idea here is that if we have a resin that has beads in it, and of course this is shown not to scale. The beads are very almost microscopic, but the proteins that are very large, like this one, volume between the beads. But the beads have these channels through them, these very small channels, and so the big proteins can't enter that. However, if we have a very small protein, let's say, this could then enter these channels and go through the beads, and it would experience a greater volume as a result. So what that means, right, the beads. So they're going to only travel around the beads and experience a smaller volume, and then they will migrate faster through this type of column. And so it might look something like this. If we have our mixed protein solution here with different size proteins, and we add that then to our gel filtration column, the large proteins, as we flow more buffer over this, those large proteins are going to migrate through faster, and they are going to elute first, whereas the smaller proteins, because they can enter those channels, are going to migrate later. They're going to migrate more slowly through the column. There are a couple important terms I want us to understand surrounding this, and one of them has to do with the idea that around the beads, in both of these cases here, there's something we would call the void, right? If you think about the beads, they're spherical, and so they're not going to pack 100% of a given volume. There's going to be space around the beads. That is called the void. And remember, these large proteins, the very large things that cannot enter any of those channels, they're only going to experience the void. And we call that the exclusion volume. because those large things are excluded from entering the beads. And so we also might hear this term the void volume, and in an ideal situation the void volume and the exclusion volume are the same thing. Now that's in contrast to the small proteins. The small proteins can enter the beads, and the very smallest proteins can essentially enter all of the channels in here, and so those are going to experience the inclusion volume because they're including the volume that's within the beads. So we're going to definitely have to understand the inclusion and exclusion volumes here. Now, in addition to purification, we can use gel filtration or size exclusion chromatography to determine the size of proteins and possibly quaternary structure if we know enough information about the system. So for example, let's look at an elution profile for a gel filtration experiment. So we can look at concentration or amount of protein as a function of, we could say, the volume or time or fraction number, whatever we happen to have. And let's say we ran some standards, and let's say our standards were proteins 1, 2, 3, and 4. And let's say I've listed them in order of decreasing size. So the elution profile might look something like this. Protein 1 will come off first, then protein 2, then 3, and then 4. So this is 1, 2, 3, and 4. And then let's say we also have some unknown. Now we probably know what the protein is, but maybe we don't know what the size is. Let's call it protein A. Or maybe we don't know if it forms a monomer or a dimer. And we then in another run that protein through the column by itself, and we get this elution profile. So maybe it comes off there at this position. So that's protein A. Well, we can use, because these standards have known masses, known molecular weights, and we know the volume they eluted on, we can identify what those volumes are, and we can know what those molecular weights are, and we can do the following plot. If we do a log of the molecular weight of those standards as a function of the volume, the elution volume for them, we would get a plot that looks like this. So let's think about this. The protein 1 would come off first. It's going to be a larger protein, so it's going to be up here. And then protein 2, 3, and 4. This should form a straight line. And that line will have equation, of course, y equals mx plus b. And we can solve this. We know then what the slope is and the intercept and all of that because we know both the volume and the log of the molecular weight because those are standards. What we can then do is take the elution volume for A and plug that into the equation for x, and we can determine y, the log of the molecular weight, which we can, of course, then determine the actual molecular weight in grams per mole or kilodaltons or whatever units we're using. So if we then know, for example, what the molar mass or the molecular weight is supposed to be for the monomer, because we use something like prot/param to determine that, then we can determine whether our protein is a monomer or a dimer or a tetramer. So for example, if we had a protein whose monomeric mass, prot/param told us that it's 45 kilodaltons, but if it was running through the column at 90 kilodaltons, that would tell us that that protein is a dimer. And indeed, this is a technique that's often used for both purification and size determination. Another technique is affinity chromatography, and this is exploiting the fact that proteins, especially enzymes, are going to bind something, some kind of small molecule that's part of its function. And if we immobilize that molecule or something like it to a bead, then the protein's going to bind that because it normally binds that, and a lot of other proteins won't. And this molecule here, for example, is a dye called cibacron blue. And this is a nucleotide mimic. If we take this cibacron blue and immobilize it onto a resin, onto a bead, then proteins that typically bind nucleotides, so ATP binding proteins or other nucleotide binding proteins, will encounter this. And notice the structure, it's kind of got these planar aromatic groups on here that sort of mimic the nitrogenous bases. Those nucleotide binding proteins will have high affinity towards this and bind to it. And so an experiment might look something like this, where we have, again, our mixed protein solution, and we add this to our affinity column, our affinity chromatography column, and some of those proteins are going to have no interaction with that ligand, and so they're going to go right on through. But some proteins will bind to it like this, and we can elute those proteins off the column with something like high salt to interact or interfere with the interactions there, or other compounds as well. And so here has been a very effective way to separate that. Affinity chromatography tends to be a very, very good way to separate proteins from other contaminants. Now, we are not going to use this in Unit 1, but we will utilize this later on in Unit 2, and we're going to utilize an interesting affinity that occurs between nickel ions and the side chains of histidine residues. And so we'll explore that in Unit 2. Another thing that we're going to do is dialysis. This is a way to filter out undesirable components or exchange buffers. And in a dialysis bag shown here, we'll have very, very small holes. And so things like salt and buffer molecules can get on through, and of course water can move on through those holes, but proteins are too big. Proteins cannot get through those holes. And so we use dialysis for a few reasons. We might use dialysis as a first step to remove some contaminants in a protein preparation. We also might utilize it to change the buffer. And this is what we do quite a bit in biochemistry, and it's certainly what we're going to do in this class. We're going to utilize dialysis particularly to change the buffer. We're going to want to change the salt and also the pH for experiments that we're going to do. And this is a very efficient way to do that by just having the desired buffer here, and that will slowly diffuse in and out of the dialysis tube. And then a lot of times what we'll do, just to be safe, if we want to really ensure that we have reduced or increased the salt concentration or changed the pH as we wanted to, we'll actually take this dialysis bag out and repeat the process and put it in a fresh new beaker or tube of buffer to ensure that we really have changed the buffer to the desired, Finally, I want to talk about purification table, and there's more information about this in your course pack, but a purification table is a summary of a purification scheme, and it gives us important values that really show us how have we done, how well have we purified the protein. And so we would look at things like these different purification techniques, like where do we start with the lysate, how well did ammonium sulfate, and then ion exchange chromatography and say gel filtration chromatography, how well did those processes purify things. And so we would report things like the total volume of our protein sample, and that volume may go down, it may increase, it kind of depends on what we're doing. We also might report the total protein. And remember, the goal of a purification is to get rid of the contaminant proteins. And so notice that as we descend this list, go through these different procedures, we actually reduce the total protein, which makes sense. We want to get rid of those contaminants. Hopefully we've gotten rid of those contaminants. And the way to assess that is to look at the activity of this. What's the total activity present in the sample? And do notice that that does decrease over each period, or each protocol, each method. And the reason there is we are probably losing a little bit of our target protein, of our beta-lactamase. Now, hopefully, though, we lose more of the contaminating proteins at the expense of a less pure sample. So the thing we're going to use to really explore the true sort of purity of the sample is this thing called specific activity. What we want to see is that as we go through the process, that this increases. Now, the specific activity This measures the portion of a protein sample that this increases. Now the specific activity, this measures the portion of a protein sample that's catalyzed that sent to hydrolysis reaction. In other words, it's the amount of product formed in a given amount of time divided by the concentration of total protein in the sample. And as we saw with assignment 4, we introduced this thing called a unit, and that is a micromole of product produced per minute. So that's an activity, a rate of formation of product over time. That's what activity is. And so dividing that activity by the total protein in the sample, that gets us this thing called specific activity. And that's purity, right? As we go along the process, hopefully we're retaining getting a much purer sample in the end with a very high specific activity.